Again, good morning and welcome, my dear medical students. Up to now, we have seen the three types of muscles, differences between all three types, and basic properties of the all three types, or I can say the muscular tissue of the body. We have seen the contractile tissue in the form of the muscle proper, then myonemes or fasciculus, that is a bundle of muscle fiber, and the muscle fiber also known as myocytes or the muscle cell. So this is the basic pattern of or basic structure of a cardiac muscle. Now this is the example of muscle which is covered by the epimysium. Then the fasciculus or myonemes are covered by the perimysium, and the muscle fiber proper or the muscle cell or myocyte is individually covered by a delicate connective tissue layer known as endomysium. Now I will explain the muscle fiber or the myocyte proper. What is a myocyte or a proper muscle cell? A muscle cell or the muscle fiber we can see here the section of a muscle. Here we can see the section of muscle which is formed by the collection of the fasciculus or the myonemes. This is the muscle, skeletal muscle, which is having the transverse striations so also known as the striated muscle. Proper muscle is formed by collection of myonemes or the fasciculus, and it is encysted by the epimysium. Here I will draw the fasciculus, which is again formed by the collection of the myofiber or the muscle cell or the myocyte, and it is also transversely striated. So this is the fasciculus or bundle of the muscle cells or the myofiber, and it is covered by a tough connective tissue layer known as perimysium, and it is having the muscle cells or the myofiber, and the muscle fiber. This is a muscle fiber or the muscle cell. This is the myofiber or muscle cell, and it is covered by a thin, delicate sheath of connective tissue known as the endomysium. And the covering membrane of the muscle fiber or the myofiber is known as sarcolemma. So this plasma membrane is known as sarcolemma, and the plasma of or cytoplasm of this muscle fiber is known as sarcoplasm. Why the muscle is having cross striations? Why this muscle or this fasciculus is having cross striations, or I can say the dark and light areas, alternate dark and light, dark, light, dark and light areas. This sarcoplasm or the protoplasm of this muscle fiber is having longitudinally running myofibrils. It is having longitudinally running myofibrils so I can say the muscle is formed by the collection of fasciculi, and fasciculi is formed by the collection of the myofibers or the collection of muscle cells, and the sarcoplasm or the protoplasm of this muscle cell or the myofiber is known as the sarcoplasm, and this sarcoplasm is having the longitudinally running fibers known as myofibrils. So these are known as myofibrils. Here I will draw the diagram of a myofibril. This myofibril is formed by the contractile proteins known as actin, myosin, tropomyosin and troponin. So the microstructure of this myofibril is having the myofilaments. This is having, these myofibrils are having myofilaments. Here I will draw the diagram of a section of myofibril, or I can say the part of myofilament. This is myofilament, and collection of myofilaments is forming the myofibril. So here we can see the this is the basic microscopic structure of a myofibril, which is formed by the myofilaments, which are the proteinous contractile filaments. This part is known as the I band. When we see this part in polarized light in the microscope, it looks light, so this part which is light is known as I band, or also known as isotropic band or the light band. So isotropic band or light band is having the thin filaments, and these thin filaments are formed by the actin, tropomyosin and the troponin, and this comparatively darker part, when we see the myofibril in polarized light in light microscope, this part, comparatively darker part, is known as A band. A stands for the anisotropic, band, and the median part or the central part of this A band is comparatively lighter, and this is known as H zone. So this part, which is comparatively lighter, in the central part of this A band, this is known as H zone, and again the central most part of this H zone is comparatively darker, so this is known as M line. The central part of H zone is comparatively lighter than the rest A band, and the central part of this H zone is comparatively darker and known as M line. Now, the part of I band or the light band, which is comparatively darker, This is known as the Z line or the Krause's line. Z line, or also known as Krause's line, or sometimes also known as the Krause's membrane. Listen again. Myofibril, there are hundreds of myofibrils embedded longitudinally in the sarcoplasm. So I can say the muscle fiber, muscle cell or myocyte is formed by the collection of the myofibrils. Each myofibril is formed by the arrangement of the myofilaments in a alternate dark and light parts. The light part is known as the I band or isotropic band, and the dark band is A band or also known as anisotropic band. This A band is formed by the myosin, also known as thick filaments. So the myosin is thick filament, or I can say the thick filament is formed by the myosin. On the other hand, the thin filament is formed by actin, tropomyosin and the troponin. These all are the contractile proteins. So the dark band and light band. The central part of dark band is comparatively light and known as H zone, and centralmost part of H zone is comparatively lighter, comparatively darker and known as M line, and the part of or the transverse lines in the light band or I band are comparatively darker, known as Z line or the Krause's lines, or also known as the Krause's membranes. Now, the part of myofibril, between two Z lines is known as sarcomere. Sarcomere is the functional part of the myofiber or the muscle cell. So this part between two Z lines is known as sarcomere. This part is known as sarcomere. So why the sarcomere is the functional unit or the functional part of the cell of muscle or myocyte or myofiber? When ATP and calcium are released from the sarcoplasmic reticulum or reticulum, sarcoplasmic reticulum releases the calcium and energy is provided by the mitochondria by the oxidation of ATPs. When the energy is released in the muscle, there is sliding of the actin or thin filaments between the thick filaments. There is sliding inside from both sides. So there is no individual shortening of these thick fibers and thin fibers. Listen again. There is no active shortening of the thick and thin filament during muscle contraction. there is sliding of the thin filaments inside the thick filaments during the muscle contraction, and this sliding of the thin filaments inside the thick filaments is responsible for the muscle shortening lengthwise during the contraction. So this is the basic functioning of a muscle during the active contraction. The myofilaments, the thick and thin fibers, never shorten during active contraction of muscle. There is only sliding of the thick filaments in the thin filaments, and when this muscle is in contraction stage, we can see this part is more darker during muscle contraction due to overlapping of thin and thick filaments in the central part, or I can say in the H zone of the muscle, which is comparatively lighter, but during contraction it becomes comparatively more darker. So this is the mechanism of muscle contraction. Now I will move on the fascicular architecture of the muscle. Muscle fiber is having average length 1 millimeter up to the 300 millimeter, or I can say 30 centimeter, but it may be up to 600 millimeters, or I can say 60 centimeters. Or 60 centimeter, and the diameter of the muscle fiber is 10 to 100 micron, and it is covered by the endomysium. Collection of muscle fiber is forming the fasciculi, and the arrangement of fasciculi is different in different muscles according to pull, force, and the work done by the muscle on a given joint or a particular joint. So basic fascicular architecture of the muscle. There are different types of arrangement of the muscle fibers or the bundles of muscles in different muscles. There are different types of architecture. The parallel muscle architecture. It depends on the force generated by muscles and range of movements. So parallel type of muscle are having the maximum range of movement. The muscle acts on a joint. Here is one muscle, and this is the joint. There is fleshy origin of muscle, which is attached to the periosteum of the muscle directly, and muscle is attached on the bone of other side of the joint by a tendon. Tendon is a connective tissue. This is the myotendinous junction. Here is the myotendinous junction. So, the parallel type of muscle arrangement, we can see this type of arrangement, that is parallel type of arrangement in the different ways. It may be quadrilateral, like in the thyroid muscle. The muscle which is extending from the hyoid bone here to the thyroid cartilage. The oblique line of the thyroid cartilage is the thyroid muscle, and this is having the quadrilateral muscle, thyroid, or I can say here is the hyoid bone which support the tongue above and the larynx below. So lower part of hyoid is having larynx and upper part is having the tongue. So this encourages the larynx by some muscles. This muscle is thyroid, and this is the quadrilateral muscle having the parallel arrangement of the fasciculus or fasciculi. The arrangement here is quadrilateral. In some muscles, the fibers are parallel, but muscle is strap-like. We can see the parallel muscular fasciculi and the strap-like muscle in the infrahyoid muscle. Infrahyoid muscles. The infrahyoid muscles are sternohyoid muscle, omohyoid muscle, the sternothyroid muscle, and this type of pattern is also seen in the sartorius muscle. Extending from the hip bone, crossing the hip joint and also the knee joint, a long muscle also known as Taylor's muscle, due to flexion of the hip joint and flexion of the knee joint. So strap-like pattern. Here the pattern of muscle fibers or the fasciculi is like this, long. So this is the strap-like pattern of the parallel muscle fasciculi. Many times or sometimes in the body, the pattern is strap-like, but it is having transverse tendinous intersections. The muscle is strap-like, but it is having the transverse tendinous intersections. So I can say the strap-like with tendinous intersections. This type of arrangement is seen in rectus abdominis. Example of this muscle is rectus abdominis. Sometimes the parallel fibers are in a fusiform shape. Here we can see the arrangement of fibers like this. This is seen in the biceps muscle of the flexor compartment of the arm. This muscle is an example of parallel arrangement of the fasciculi in a fusiform shape. This type of pattern, which is seen in the biceps brachii, is also seen in a muscle of the head and neck known as digastric. This is having anterior and the posterior belly of digastric. Both bellies are having fusiform arrangement or shape of parallel fiber. Here is a fibrous pulley which encourages the muscle to the hyoid bone. So this is the posterior belly and here is the anterior belly of the digastric. So the parallel architecture of muscle fiber is of different types. They may be quadrilateral as seen in the thyroid muscle, may be strap-like, which is this pattern is seen in infrahyoid muscles and the sartorius muscles, and the strap-like but having tendinous intersections. This type of pattern is seen in muscle of anterior wall of abdomen, that is rectus abdominis muscle, and the fourth pattern is the fusiform pattern of parallel fiber, which is seen in the biceps brachii muscle and the digastric muscle. So this is the parallel architecture of the muscle fiber. Now I will talk about the second type of architecture, that is the convergent architecture.